Next, we'll be moving into the receptor tools work, and Eve will be presenting on TCR match and then a little bit of BCR match at the end there. So thank you so much, Eve.
Yeah, so yeah, thanks very much, Nina. As Nina said, I'm going to be finishing up on the receptor tools, focusing on our sister tools, TCRMatch and BCRMatch. So I'll sort of give you an overview of how to use them and how they work. And yeah, I think they're very useful tools that hopefully people can make good use of. So obviously we've just recently heard from Lonneke, who gave a great overview of adaptive immune receptor biology and how yeah, the sort of the anatomy of the adaptive immune receptor. So TCR match, this works on TCRs, BCR match, that works on BCRs. So obviously the T cell receptor and the B cell receptor, they're both these dimeric immune proteins that are formed from somatic recombination. Each of the chains within the receptor having three complementarity determining regions, as you can see from this diagram. Um, TCR match has this special focus on the CDR3 beta. So, like with BCRs, there's this concept of the specificity determining CDR3, where you have this hypervariable loop that spans the recombined V gene, D gene, and J gene. So it's more variable. And also specifically for TCRs, canonically, the CDR3 beta is the part of the TCR that's most involved in recognizing the peptide as opposed to the MHC molecule itself. So as a result of that, TCR match focuses on the CDR3 beta and doesn't use these other two CDRs. Um, so very common experiment now that we do another view into how the immune system works is repertoire sequencing. When you perform repertoire sequencing, you can get tens to millions or tens of millions of TCR sequences. So in this example, we've got these six CDR3 betas. The limitation is that TCR sequencing by itself, with the exception of certain experimental setups, doesn't directly reveal the epitope that's recognized by a given CDR3 beta. So you have all of this data about which TCRs a person is making, for example, but you're not able to map those to the epitope specificity. So the question is, how do we know what the epitope recognized by a TCR is, and how can we best make a prediction in any case? Uh, so I think that the best thing to do is always to go to the data. So going to the data first, as Lonneke went through, we do have a substantial amount, particularly of T cell receptor data, but also of B cell receptor data. The number that Lonneke has just presented is nearly 190,000 T cell receptor sequences. So if you have a CDR3 beta of a T cell that you're interested in, it's absolutely worth looking for exact matches within the database, and there's a good chance that you'll find that exact CDR3 beta in the database with so many. Um, and so that's the general concept of TCR match and of BCR match. So it's using the sequence of a known receptor to match your query. So this example, if we take this first CDR3 beta and look at the IEDB, it may be that we find an exact match. So this is the case here. So we found it, we've looked in the IEDB and we found something that's 100% identical. And then from that, we can make the mapping to the epitope. So in this case, I think this is an epitope of some protein in SARS in a beta coronavirus. But it may be that we have a slightly different CDR3 beta. And so if we're looking just by exact match, we're not going to find anything. And so we're unable to make a prediction. And that's when we start considering things beyond an exact match. So how similar does a CDR3 beta need to be in order for us to confidently make a prediction that it shares an epitope? And so all of this work, if you're more interested in the specifics of the various metrics that have been explored, this is all available in the original 2021 TCRMatch paper. But these are various ways where one could make a mapping between one CDR3 beta and a slightly different CDR3 beta. Of course, when we make any kind of prediction, we're operating in this space of having a trade-off between precision, so making as few false positives as possible, and our recall or our sensitivity. So as you move along the recall space, you'll have a decline in precision, and so that's sort of the consideration to make when you decide how dissimilar a CDR3 beta can be, and you still use it in your prediction. So in the TCRMatch paper, these values were calculated at a range of thresholds for all of these different metrics. So the alignment score, identity, the Levenstein edit distance, and then we have our method, TCRMatch, and then there's this additional method, TCRdist, which has since been updated actually, sort of a conceptually similar thing of having some weighted mismatch distance between the two sequences that extends beyond identity alone. And so you can see where we are where you want to be, which is above the identity metric, i.e. for a given sensitivity, you are more precise, i.e. you're making better predictions effectively. So that's the TCRMatch, and that's where it compared at the time in 2021. So now I'm going to talk more about how TCRMatch actually works and how you can use it on the classic tools site. So TCRMatch is a comparison of all of the Possum k-mers using Blossom 62. This is an example. So we have these two CDR3 betas. Something to note is that they're not of the same length. You've got a length 10 one and a length 11 one. And that's a nice advantage of using this k-mer approach. You don't have to have length matching, and that allows you to increase your sensitivity and find more dissimilar matches. So what is this comprehensive k-mer matching approach? You take each of your CDR3 betas, you'll select a k value for your k-mer. So we'll start off with 1. And then for each of the pairs of k-mers that are possible within those CDR3 betas, for example, these equivalent positions, which are unchanged, we'll take the associated value in the Blossom 62 matrix. In this case, this is a 4. And we'll do that for each of the possible comparisons. So obviously an alanine to alanine, so non-transition, that's going to be more favorable than an alanine to a serine. And that's more favorable than an alanine to an isoleucine, and that's more biochemically favorable in terms of the chemistry of the amino acids. And so we continue that on and on. I won't make you look at all of the possible k-mers, but we will start off with k equals 1, and then we move to consecutively from k equals 1 to k equals 10. So for the k equals 2, what we would do is we would take the both amino acids, we'd look at the alanine to the alanine, which is a 4, the serine to the serine, which is a 4, and we take their product. And we'll do that for all of the possible k-mers. We'll multiply the substitution values, and so on and so forth. And so on. So then we keep going, we keep increasing the k values from 3 to 4 to 5, all the way up to the length of the shorter CDR3 beta, which in this case is 10. So we would have only 2 possible k-mers there, this one, and then with a shift of 1. And so you end up with a value for each of the possible k-mers. You sum them, and then in order to get a score between 0 and 1, so that you can compare that to an exact match, we use we normalize by the square root of the TCRMatch score for identity to oneself for each of the CDR3 betas. And so in the end, we end up with a score between 0 and 1, where 1 would be the perfect match, which would be an identical CDR3 beta. So hopefully you understand a bit more about how TCRMatch actually works. And now we can, I'll just show you what the website looks like in terms of the input and the output. So in terms of the input, you can put your CDR3 beta sequences in as either a FASTA file, new line separated file, or a space separated format in this box. You also, if you have sort of too many, or it's more convenient to use a file, you can upload a file, although that can't be larger than 500 sequences due to the computational cost associated with TCRMatch. You can also upload the Trust4 input from a file, so that was a relatively recent addition. So you can take the output from Trust4 and feed that directly into TCRMatch. Another thing that you can select is the database. So you can use the IEDB. You can also use CEDAR, the CEDAR database, which is our cancer database. And then finally, you have a question of how confident do you want your predictions to be? So the recommendation is a score of 0.97, and this was from based on the original paper, where that 0.97 threshold gave the maximum precision of around 70% at a recall of I think it was 8%. So obviously increasing that value, you'll be more confident in your predictions that they're correct, but you'll get fewer of them. So you might miss out on some interesting things. So this is what the output looks like. You have a column for your input sequence. Then you can find your corresponding matching sequence in the database alongside the score. So you can see here that's an exact match in this top one. Um, there will be a list of the epitopes that are associated with that particular CDR3 beta. So as you can see here, there can be many, as well as the corresponding antigen from that epitope and the source organism for the antigen. So one thing to note is that this is also available as a standalone tool. So if you'd like more flexibility, so say you have your own database that you want to match against, you can download and use a standalone tool, and that will give you full flexibility. So as we get to the end of the TCRMatch section of the presentation, I'd like to just briefly acknowledge all of the contributors to the original work. So here's a paper. This was published in 2021, as well as Raphael, who has been the developer and maintainer for this tool over the last few years, and has made some interesting some valuable contributions towards continuing to improve TCRMatch as we get more data. So having spoken about TCRMatch, I'm now going to move on to BCRMatch. So as I say, they're sister tools. They are trying to do the same thing of predicting receptors that bind to the same epitope. So BCRMatch is currently only available as a command line tool. It will be added to the next gen tools, but as a command line tool, it uses pre-trained machine learning models to predict if a pair of antibodies share the same epitope. So one difference from TCRMatch is that the CDRH3 isn't sufficient, and that was actually a really interesting finding from Mahita's paper, where she introduced this method, that there's really valuable contributions from both the other heavy chain CDRs and also from the light chain. And this is a really interesting area where we've seen over the last couple of years how much of the contribution to specificity comes from the germline encoded regions. So all of that information is used by BCRMatch, and as a result, it requires sequences from all of the 6 CDRs. If you only have full-length sequences, the tool will extract the CDRs for you. So as I said, this uses machine learning models. Those are pre-trained and distributed as they were in the paper. So they're trained on antibody pairs that from the IEDB or from additional data sets from previous papers. And those are antibody pairs experimentally validated to share an epitope. And so, as I said, this work is available as a preprint. So there's some really interesting findings from that about um, our understanding of how specificity is encoded in BCR sequences. So I'd really recommend reading this. And there's also additional data about how BCRMatch was trained. So in the last few minutes, I'm going to talk to you about precisely how BCRMatch works. It works on a pairwise basis. As I said, it also uses all six of the CDRs. So here's an example, two antibodies from the PDB. So you would basically you match up the CDRs, and for each of the pairs of the CDRs, you calculate a KMA score. And that KMA score is calculated using TCRMatch. So that gives you then, for each of the six CDRs, a score that denotes their biophysical similarity. This length 6 vector is then used as the input to a machine learning model that was trained on the IEDB. And the output of that then is this is a score. So there's multiple models. It was 5 models in total that Mahita explored in the BCRMatch work. The ones that are recommended are the logistic regression and then this Gaussian naive Bayes model. But each of these models will produce a different score. Then the tool ranks those scores in order to make it more robust against the scores that were observed from the training data. So you'll get a percentile rank for each of the models. And then to make it even more robust, those scores are used to calculate a harmonic mean across different machine learning models. So the total effect is that you're ensembling, which is generally a really great method within machine learning to improve performance and to improve robustness. So this isn't currently on next generation tools. This will be part of the BCR tools release, which will happen early next year. But here's a little sneak peek as to what BCRMatch will look like when it's up there. So again, you'll have your input. In this example, we've got like a tabular input for each of the CDRs, but there will also be the option for full length sequences. And then you're able to select certain of the prediction parameters. So an interesting one is you can select the different training data. So you have the IEDB data, and then you have this data obligity. So this is a previous publication that was trying to do a similar thing. So that you may get slightly different results. So we recommend the IEDB as a default. You can also select among the different prediction models from logistic regression to XGBoost, random forest. Um, so yeah, there's various different prediction models that you can use. And then the output will be for each of the pairs that you put in. So if you imagine like a matrix of where the rows and the columns are the antibodies you put in, this will calculate for the upper triangle of that matrix, all of the pairs, a prediction for the models that you selected, as well as their mean percentile rank. And you can use that in order to rank your pairs of antibodies according to how likely you think they are to bind to the same epitope. So that's all I've got. That's all I've got for you on TCRMatch and BCRMatch. I'd be really happy to answer any questions. And yeah, just finally would like to acknowledge Mahita, whose work this is, that you can read about on bioRxiv. And thank you for listening. I think I'm on time. So yeah, happy to take questions now. Thank you.
You are perfectly on time. Thank you so much, Eve, for that wonderful presentation. And it's great to hear about the work that other members of the team have done as well. And thank you for acknowledging them too. But so that was awesome. And now we'll move into our last Q&A section of the day. So I'll invite Lonneke back to join Eve. And Lonneke, while we give Eve a minute to go through the questions, I'll let you take the first one.
Yes, so I'll start with this question from an anonymous person. Do you have a hierarchy in assigning the gene names, such as tidy T cells over IG blast? So this is something that we have that I have looked into a lot, like because I have for each receptor all these um, different suggestions of what the gene names would be by different tools. The ideal gold standard is always if we have the full nucleotide sequence, we can get the most precise annotations, and then we will run IGBlast nucleotide version and use those VDJ gene names. So that's usually the best we can get. But often we don't have the full nucleotide information. And still, if we have full amino acid sequence information, we would still, for the V gene at least, we would use IGBlast on the amino acid sequence and then prefer that V gene as the next best option before we use the tidy T cells version. And the reason for that is that maybe the tidy T cells, it doesn't do any, the only thing it does is just correct the name. It corrects what the name looks like. So really, these are the reported gene names. But maybe those gene names were reported a long time ago when we didn't have the current reference sets available. So with the updated reference sets, we would prefer we seem to have better results with IGBlast amino acid than with using the curated values. But for the J gene, we don't want to annotate that based on the amino acid sequence because it is too short. So there we just use either IGBlast nucleotide or whatever the tidy T cells standardized value of the curated value would be. So yeah, it's a bit different for V and J.
Okay, great. Thank you. I'll move on to my first question. So while the details of TCRMatch are fresh in our minds, is it possible to change using a different Blossom matrix for analysis, or Blossom 62 is fixed? Thanks. That's from an anonymous attendee. So yeah, great question, and this is a really cool research direction. So for example, TCR-DIST have done an update where they've used a custom substitution matrix. So if you would the best way to do that in practice would be to use the command line tool version of TCRMatch. And you can't, unfortunately, you can't trivially specify a substitution matrix, but you can, that's something you can modify in the source code. So that would be the best way for doing that. But yeah, unfortunately, you can't currently supply a substitution matrix as a command line argument, for example.
And I'll take the next one. So what is the species coverage like for receptor data? So on the TCR side, it's quite simple. The vast majority of the data is human data. And then we also have some, I think a few thousand records of mouse data. But I don't believe we have any other species represented on the TCR side. With the BCR data, it's a lot more diverse. So still the majority is human data, and then the second most represented is mouse. But we also have quite a few antibodies from like llama or alpaca and other kind of model animals like macaque. And like earlier when I was looking into this, I'm just now pulling up for myself my old slide, which has a whole list. There's maybe like 20 different animals where we just have 5 or so receptors in the IEDB. So yeah, it's really the majority human and mouse, and then a few of these other model species that have like a substantial amount, and then a lot of species where we just have a few that happen to have been reported at some point.
Great. And then I'll just answer this question from an anonymous attendee about, is there much IEDB data overlap with VDJ-DB? So absolutely. So we are doing very similar things. So there is a very substantial overlap. So in the Venn diagram, we each have something that the other doesn't have. So if you're trying to generate the most comprehensive database possible, for example, for training machine learning models, it's great to use both VDJ-DB and the IEDB.
And then I will take this one, which is also from an anonymous person. What reference sets do you use for the BCR and TCR genes? And so, as you maybe you are aware of this database called OGRDB, which is a air community, like a community sourced reference set of immune receptor genes. So wherever possible, we will use OGRDB. But right now, they only have for BCR reference sets available, and the TCR reference set is not yet available. So there we are using IMGT. But yeah, it also should be kind of noted that it's uh, it's only when we have full sequence information available that we can use these reference sets, which a lot of the time are it's only a minority of the data, except BCR amino acid, of course. But yeah.
Great. And then I will answer this question from Pramod. Is it possible to run TCRMatch on an entire repertoire? So yes, there is significant computational cost associated with running it on I'm thinking of an entire repertoire in terms of tens or hundreds of thousands of sequences. It may be the repertoire is only 1,000 sequences, in which case that's something that is relatively trivially done. You can do that in two runs on the web server. Um, when I have run TCRMatch on large repertoire data sets, definitely using the command line tool and using parallelization are the best ways to get that done on a reasonable timescale. And VDJ server also has been hosting TCRMatch on there, so that's like an additional source of computational power should you need it for running this tool on an entire repertoire.
So then I will answer, why is there so much more TCR data than BCR in the IEDB? And so the IEDB is just reported what is being published. So when there is this kind of discrepancies in what is reported in the IEDB, it's because there's different numbers of data that are being published. But one thing that also we should take into account is that IDB, we are publishing data that where the specific epitope is known. And so there exists a lot of BCR data where we know, for example, an antigen on a larger scale, but we don't know what specific epitope of the antigen. And so those data would not be included in the IEDB. But one of the other repositories that is part of the AKC integration project is called IRAD, and they are also storing BCR data where the antigen is known on a broader level with not just the epitope information. So so that could be another source of finding a bit more BCR data, because IDB is really everything that is epitope specific.
Okay, great. And I think, okay, there's one that is that I can answer. So a question from Josh Della Vedova. In the data used to train BCRMatch, how do you determine whether two antibodies bind the same epitope? Great question. So there's two big training data sets within the BCRMatch paper. So there's one that I referred to as that's referred to as the Abligity data set, and that one was sort of defined in a custom way where they had a biophysical similarity metric called Abligity that they ran on crystal structures, and so they determined that based on the physicochemical similarity between the epitope recognized in a pair of antibody antigen structures. The IEDB dataset that was based on the exact epitope label as provided in the IEDB. So the epitope, the discontinuous residues that are part of that record, as well as the source antigen. So that was used for the designation of antibodies binding to or not binding to the same epitope. And so, the total data set for both of those, I think for Abligity was upwards of 300,000 IEDB, I think 150,000, and a really small fraction of that is actually positive.
All right, I will take this one. How did you validate the curated CDR3s? So for all the curated CDR3s, we are running this new functionality in tidy T cells where we are aligning the CDR3 to the VNJ gene information. So if a curated CDR3, if the names of the VNJ genes were also provided, we will look up what was the sequence of those VNJ genes and try to see if it matches up with the CDR3. So that's how we would validate them. Validation would then be like, okay, we don't need to correct it, but we see that it lines up with the genes. And if the VNJ gene information is not available, then we just align to all available genes. So it will take a bit longer. It's a bit more computationally expensive, but that's how we can tell apart an actual CDR3 sequence from a sequence that is complete gibberish.
Great. Let me just look. I think I've said that I'll answer this one, but this is your realm, Lonneke, so feel free to correct my answer. But which data format would you recommend for the downstream use with external analysis tools? For example, the TCR Explore R package, and that's from an anonymous attendee. So I'm not familiar with the TCR Explore R package. So having had a quick look, I don't see mention of the air format, but more generally, I would definitely recommend using the air format because multiple software tools go to the go to considerable effort in order to be able to natively support that data format, and even just more generally, it's a convenient, very well defined tabular data format. So that would be my recommendation.
Yeah, and I think I can just add on to that like in the different data exports that IDB provides of the receptor data, it's not like there is really that the information itself is different. It's really the formatting. So air format would be a different formatting of the same data that you would get from the other receptor exports. But I did very briefly Google the package that you did mention, which I believe is meant for repertoire analysis. And I just wanted to also mention that the IDB is reporting all these individual receptors that have been derived from all these individual different experiments. So the types of analysis that you would do on that data would be different from the types of analysis that you would do on data that comes from a blood sample, for example. So if I take a lot of blood from one person and I look at all the receptors that are in there, then you can say something about the diversity and on all these kind of things, and then try to infer something about the health of that person. But when you're taking data from the IEDB, it is not like a natural repertoire because it's a set of receptors that were derived from different experiments. So you have to be a bit careful what types of tools you would use this data on. It's really more a set of receptors than a repertoire from a person, so to say. I can also just move on to then this question of when will the new tidy T cells be released? So this is a collaborative effort. I really hope that it will be kind of released in let's say the spring at the latest. But yeah, no promises.
Awesome. Thank you both so much, Eve and Lonneke, for not only the wonderful presentations, but the really in-depth responses to the questions as well. And it looks like we are about done. So thank you both so much.